Something is wrong. Okay, this first slide shows you that to develop a new drug in the cardiovascular field is a complex, well, it's a complex because as you can see here, and probably you have seen this slide many times. This is our own slide because we came from 10,000 compounds. At the present time, it's not 10,000, probably it's 50,000, 60,000 compounds. Oh. And as you can see, during a period of approximately 12 years, if we are lucky enough, from 20 to 50,000 compounds. Oh, fine. One will reach the market. Okay? Now, if you look at if we have been very successful, this is 1998, this is 2017, and please note that we can set that there is a line, what we call the line of 30 compounds per year have been approved. And this is a surprise because I thought that with the new genetic knowledge, the new technological developments, we will have more and more compounds. It's true that the number of compounds in red are increasing. This red color represents biological compounds. So it's a long process. It's inefficient if you want because we did not improve the number, and it's pretty expensive. And this, the Tufts University in the United States, they perform very specific studies on drug development and marketing. And as you can see, between 2003 and 2014, you can see there's a marked increase in how much money we must invest to develop a successful compound. But this is the most important one for me. This is a paper taken from Nature. And what you can see is that if we analyze why new compounds cannot reach the market, and we analyze phase 2 and phase 3, it's really it's a surprise why we reach a phase 3 trial to reach the conclusion that 50% of the compounds are out of the market because they do not have efficacy. Oh my God, why we reach the phase 3? Why we don't stop this in phase 1 or phase 2? And please note that 35% of the compounds go to the basket, not because of efficacy, but due to problems with safety. And this is really very disappointing because it cost, all this development cost a fortune, and at the end, we discovered that the compound is not efficacious, is unsafe. So this is one example. This slide has 2 years. We published the more recent one in December last year. These are several compounds that have been were developed and they fail just for the treatment of acute heart failure or chronic heart failure. Please note that the problem is not that they are ineffective. No, the problem is that not only were ineffective, but many of these compounds, for instance, this one increased cardiovascular events, this produced hypotension, this increased thromboembolic events that increase the risk of heart failure. So we develop a compound for the treatment of heart failure, but produce heart failure. Oh my God, what we are doing, no? See, this one compound, everybody remembers this compound, rolofilin, that produces seizures and stroke. Sildenafil may impair left ventricular contractility. And please, just to remind you that this study was to demonstrate that was effective in patients with with right ventricular heart failure, or tolvaptan with hepatotoxicity. So it's time to sit down and to analyze why we are failing so frequently. And probably the first point, the explanation is in this paper that was published in 2016 in the European Heart Journal. You see, the first reason is that drug therapy requires long-term treatment, years, to effectively modulate cardiovascular diseases. Many research programs involve event-driven studies where the annual incidence of events is very small, but the population at risk is very large. It's typical to analyze the effect on stroke in patients with atrial fibrillation. Clinical trials are conducted in patients receiving multiple evidence-based therapies. If you want to develop a compound now for heart failure, don't forget that the patient, if the patient has only heart failure, the placebo group will receive an ACE inhibitor, a beta blocker, a mineralocorticoid receptor antagonist, probably digoxin, and probably something else. And now, on top of these 5, this is the placebo. I'm going to add the new compound. And probably this new compound, now we said it failed. But if I test this compound 30 years ago, at the time of ACE inhibitors, these compounds will be a marvelous compound. But now, it's a compound that we consider is a completely failure. We need to demonstrate incremental risk reduction, and this requires very large sample sizes. We need thousands of patients sometimes. Cardiovascular mortality and other clinical outcomes have declined in recent years. So 30 years ago, it was very easy to demonstrate something against myocardial infarction or a class 3 heart failure, because the mortality was pretty high. Now, no, this is not the case. Clinical trials are performing large, this is for me the most important point at the present time, in large unselected patients. We talk about heart failure. Which heart failure? Probably we fail in heart failure with preserved ejection fraction because the patients were not on this condition. If we talk about acute heart failure, which acute heart failure? There are at least 6 different phenotypes. Atrial fibrillation, coronary artery disease, in whom multiple pathophysiological mechanism participates. Not all are likely to be influenced by the agent that we are studying. So the concept for me at the present time is that for the last 60 years, pharmaceutical companies were interested in developing compounds for everybody. I have this compound for all the people with heart failure, with all the people with atrial fibrillation, with all people with, and this is not true. We know that there are different pathophysiological mechanisms, different phenotypes, and we need to develop compounds for some particular subgroup of patients. Of course, this is against the dogma of the pharmaceutical companies, because they want a compound to be to occupy the whole market, but this is not. So there are no drugs for all patients. And of course, the main problem is that it's a very complex structure. I'm going to deal with this. There's a lot of bureaucracy, complex and contradictory, national, local regulatory burdens. This is typical. We try to develop a multinational company in Europe, and you found that the different countries have different rules. And even in my country, for instance, we have 17 different administrations. But there's something else that's not included here, and probably is the most important, together with this one I already mentioned. We have been very successful. We have beautiful compounds for everything, for almost all the cardiovascular diseases. And so the problem is that we have been so fantastic in our drug development that, for instance, I'm not going, just let me, probably there's something here wrong. Can I go back? Back. I thought I'd have one. No. At the back. No, you are going in the wrong direction. In the other one. Back. Something wasn't missing here. Okay, I will do it. Do you know how many compounds we have at the present time for the treatment of arterial hypertension? More than 60. More than 60. Do you know that somebody is going to invest money to develop a new antihypertensive drug? And the answer is no. Do you know how many compounds at the present time are not generics for the treatment of heart failure? 1, sacubitril valsartan. So if I now develop a new compound, I go to the administration in many countries in Europe and they say, oh, wait a minute. Enalapril is 6 euros in my country. And now you bring a new compound, sacubitril valsartan, and you say it's 100 euros. What are you talking about, man? You're crazy. I'm not going to support your new development. And so look at this. Now we develop beautiful ideas. We develop new compounds. They go to phase 2, and suddenly, not for scientific reasons, but because of commercial reasons, the compounds never moves to phase 3, or even if they move to phase 3, they never will be commercialized. one of the main points why we fail many times is because we forgot this slide. I'm doing basic science for many years, so we use animal models to determine drug efficacy and safety, but mice are not men, and a cell is not a tissue. This is something I learned many years ago. And then we moved to phase clinical trials. Phase 1 are healthy, young men. But I give the drugs. I never prescribe a drug in a healthy person. Not at all. So we need to phase 2, and phase 2 for me is very important, because phase 2 is where first of all, I confirm in patients, because phase 1 is in healthy volunteers. In phase 2, I confirm the mechanism of action that I describe in animal models or in isolated cells. I describe pharmacokinetics and pharmacodynamics, and I confirm that the drug has multiple effects. Multiple effects. I don't know a single drug with a single effect. The dose, which is the dose range, which is the best time, and what happened, how comorbidities affect the efficacy and safety of my compound. What happened? That there is a lot of pressure, and sometimes we move from basic science, and in basic science I do not have comorbidities. I do not have comorbidities. I have isolated cells, a beautiful signaling pathways. Move to phase 1, and from phase 1, we move immediately to phase 3. Hey, man, we need to spend some time here. I need to confirm the mechanism of action. I need to define the best population. There is no drug for everybody. Which is the niche? Which are the population more suitable for my compounds? So for me, first thing we need to change is we must know the drug before pivotal trials are performed. Phase 2, as I already mentioned, to understand the drug properties, the mechanism of action, the pharmacokinetics and pharmacodynamics. The mechanism of action is what we need to validate the target. Because probably, and this happens quite frequently, what I found in animal models is not present in humans. So I validate the target, or probably it's an incorrect hypothesis. And if it's an incorrect hypothesis, this compound is going to fail in phase 2 or in phase 3 clinical trials. Define the dose range, they define the off-target effects. Now, cardio-oncology. Fantastic. 1982, I remember I was working with anthracyclines. Everybody knew what was going with anthracyclines. But what happened with the new compounds? People develop compounds and look through this kaleidoscope, and they said, this is the tumor cell. Yes, and this is the signaling pathway. Fantastic. And this is my target. Fantastic. But this target is not only in your cancer cell. It's also in the heart. It's also in the vessels. It's also all around. Which is the problem? Now we cure the patients with cancer. But the patient remains with heart failure, thromboembolism, QT prolongation, blah, 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 blah. Identify the right population, as I already told you. Those most likely benefit according to the mechanism of action. We, of course, will use imaging and biomarkers as surrogates to predict the clinical efficacy and safety. We must incorporate the primary endpoints based on the mechanism of action and the right population for phase 3 clinical trials. And of course, from phase 2 up, we need to be very, very, very concerned about the possible safety concerns and about possible drug-to-drug interactions. This is something that is interesting. I'm just going to present to you that this is a sentence also from that paper that I present to you, why we fail in developing new compounds. Targeting those patients most likely to benefit from modification of receptor targets may be a more direct, efficient approach to generate robust scientific evidence and decrease the cost of failure in terms of resources and times. Dalcetracib, a compound that increases HDL cholesterol 90%. So everybody was expecting that this compound will reduce atherosclerotic diseases. Unfortunately, we found that there was no benefit at all. No benefit at all. But somebody in Canada says, I have some patients that respond pretty well. And I found a very significant improvement of these patients. He went to the pharmaceutical company. Pharmaceutical companies, I'm not interested in spending a single euro to your project because this compound was negative. But this guy, you will see the name, Caliph in Canada, identified a group of patients with a particular genetic variant in this gene, this adenyl cyclase, whatever, you know. So he came back and analyzed the patients. This is the general population. Negative result. Fine? Okay. Now he looked at this and said, look, these are cumulative incidence of events. People with this particular genotype, there was an increase in 27% in mortality. But people with this genotype, they have a 39% reduction in mortality. So if I develop a compound for everybody, this compound goes out. But of course, this is a very significant benefit. So from now on, and this is the paper from Tardif, we must think on this concept. This is the typical thing that are doing every day people working on cancer. I do not develop a compound for all types of cancer. I commercialize a compound for patients with this particular marker of cancer. Now let's see this slide. I already mentioned to you that some compounds is because there are a lack of efficacy and there are problems with safety. But please, do you remember what I told you? We have too many compounds, we were very successful. I'm not going to found you because I'm not interested in developing this compound. And look at this, business decision. This compound, I'm not interested, I know that it's effective, fantastic, but I'm not going to spend my money. But the main reason why clinical trials fail is insufficient enrollment. Okay? And here are 85% of clinical trials fail to retain enough patients, 80% of clinical trials fail to finish on time, and this cost a fortune to the people who put the money on the table. 50% of sites enroll one or no patients in their study. Usually these are the big top guys that 20 years ago signed the papers. Okay? Then I introduced 200 patients. I'm the last in contributors. Page 120, you know, in a small letter, but I'm the big guy who signed the New England. Okay? 40% of the total pharmaceutical clinical trial budgets, 40% goes to recruitment, a fortune. 30% of patients sometimes leave the clinical trial. So up to now, sponsors and CROs rely on traditional recruitment methods, like they go to the hospital, they speak with the big men, they speak with and try to, could you introduce me patients in this clinical trial? Then they go to the consumer data, go to the pharmacies, apothecas, then they select the hospital because these people is very, very important in this city and so on. Forget it. Things have changed dramatically. First of all, approximately 4 million data every day are moving through the nets, telling what? People speak about their diseases. And there is no confidentiality. They speak very freely, you know? They talk about their disease. They speak with other patients with the same disease. They talk about the MDs. They talk about the hospitals, about everything. And so there is special software analysis with natural language capacity that can identify if this particular patient that I identify in the net is suitable for a clinical trial. Patients organizations, particularly in my country, some of them are fantastic. They offer the potential to interact with individual patients, and the patients are they support what this patient organization propose. There are marketing campaigns, Google AdWords, Twitter hashtags, whatever, and there are, of course, advertisements and so on. This is one example. Here you have asthma, Compensation is 900 euros, no cost for study medication. Here, participants will be compensated for time, inconvenience, and travel expenses. I don't know how much money they are going to receive. And here, this is for diabetes, and they also offer to you benefits. This is a recent slide. This is this organization, CenterWatch, and CenterWatch found that they recruit almost 40% of patients. They don't need the MD at all. They don't need the hospital. They just used email, 60%, phone, 37%, visit in person. This is the old stuff, 3%, you know. Here you have disease-specific association sites, 14%, referrals from family and friends, 11%, only 10% referrals from the MD and the nurse. So you see how much the things changed in the last 10 years? Another problem is that clinical trials, sometimes it seems that the development was made by the devil. You know, I mean, they are unnecessary and complicated. And I always remember with my students this sentence from Lewis Samuels: complexity in a system tends to increase that system inefficiency. The greater the number of variables, the greater the probability of those variables clashing, and in turn, the greater the potential for conflict disarray. So we need to change our way. The first thing is to replace sites by consortium. And of course, this is a consortium, a group of hospitals with unified database, large banking and genetic information for present and future randomized clinical trials. High quality, secure, integrated information system among hospitals, of course, accredited. They must be accredited to reduce the multiple inspections. How to defeat the barriers, develop patient consent and standardized forms to record clinical trial data, unified database, a single review board committee per project. This is not simple, because every hospital is reluctant, because each institution brings its own values, preferences, interpretation of the privacy laws on the table, and of course requires an enormous trust. I must trust you, and you, and you, and you must trust me, you know, if I am going to be the main investigator. Another point is clinical trials generate an incredible amount of data. And many trials are still manual, oh my God, and many is written, so a lot of errors. Insecure email, I mean, I have students that can go in your computer. They can go perfectly and can analyze what happened in my hospital. Of course, this is absolutely forbidden. There will be fire. You know, but they can do it. It's so simple, you know. More than 3 hours per week hunting for clinical trial-related documents. And the number of documents are increasing dramatically. Look, 2000, 2003, approximately 106 procedures. Now are over 178. Data monitoring and record keeping comprise 30 to 66% of total costs. And of course, exchanging information via traditional, unsecure, inefficient, and not reliable audit methods is costly and time-consuming. So regulatory requirements create more urgency to exchange regulatory documents in a secure and auditable manner. And of course, what we must do is to go to the cloud. And this is just to remind you, this is improved IT trial with ezetimibe, 18,000 patients, 300,000 patients visits, approximately 2.7 million case report forms completed, 15,000 serious adverse effects processes, 30,000 monitoring visits. You can imagine the amount of work this represents, okay? So we need to go. A new landscape. We were accustomed to perform, we already moved from phase 1 in healthy volunteers, and we moved to this study, is placebo against 3 doses of this compound. We performed the study, we finished the study, we performed the data analysis, and then we start to prepare phase 3 clinical trial. This doesn't make sense. Why we don't go from here till the end? And this is one example. We start again with 3 groups, but we start to move the clinical trial, and suddenly we realize that these doses don't make sense. So we stop this. Okay? And the people from this arm will enrich arm A and C, and so on. Okay? This is what I want to show you here. This is what we call adaptive designs for clinical trials. So this seems that we can make a lot of fakes. No, 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 no. There are very, very strict rules, you know. We start the data collection according to the location and sampling rules. We analyze the available data, and suddenly we say, do we stop the clinical trial? And this arm, yes, yes, okay. Stop this arm. Or no, or we continue, and we will follow this circle, and probably you can understand much better this study. This is the first study that was performed using this idea in patients with diabetes mellitus. Of course, the patients during 11 weeks were receiving metformin. That's all. At this time, for approximately 14 weeks, patients received 7 different doses of dulaglutide, and were compared with placebo and with a typical dose of sitagliptin. During this period of time, you can see, after these 14 weeks, it was decided that 1, 2, 3, 4, and 5 doses should be avoided. Stop. So at the end, we have 2 doses of dulaglutide, sitagliptin, and placebo. And we continue the trial up to 104, 108 weeks. And we have all the information we need to be, this compound was accepted for marketing in the treatment of patients with type 2 diabetes. So this is what we call the seamless, process. Another change was that this was the typical idea we have. We develop a compound, phase 1, phase 2, phase 3 clinical trials, everything is done, phase clinical trials are ready, the drug is effective, is safe, so we knock the door of the European Medicine Agencies or the FDA and say, we want to commercialize this compound. And we receive, they evaluate the dossier, and at the end they say, yes, this compound can be commercialized. So, of course, pharmacovigilance and post-authorization follow-up. But now I'm going to propose you something. And I'm not going to develop a compound. This is typical for patients with cancer. I'm not going to develop, do you remember my comment? I don't want a compound for everybody. I want a compound for a certain subgroup of patients. If I select this subgroup of patients from the early beginning, probably I can reach the door of the European Medicines Agency, of the FDA, much earlier. And look at this. In this case, I go and ask for an initial license for something. What something? Look, this is what we call adaptive licensing. It's a prospectively planned adaptive approach to bring more rapidly promising drugs to the market. What do I mean with promising drugs? Drugs where is a medical need, orphan diseases, or emerging threats. So I receive a first indication, but I continue to study this compound, and I can later on to obtain a full license, okay? Look in the type of cancer. Opdivo. This is an old slide. I didn't change the slide in the last 6 months because there are 2 new clinical indications. Opdivo, 2014, was accepted for BRAF B60 wild-type unresectable or metastatic melanoma as a single agent. At the present time, 17 clinical indications in 4 years accepted by the FDA. Okay? That's fantastic. You see, different types of cancer. Which type of design they use? For instance, this one, the basket. We are accustomed in cardiovascular disease, I include everybody. Look, they include people with lung cancer, colorectal, ovarian, myeloma, breast cancer, various rare cancer, and they put all these people in the same basket. That's what we call the basket studies. So the basket study tests the effects of a single drug or a combination on a specific mutation in a variety of cancer types. allowing the researchers to analyze each cancer type individually. If you combine all these, you are performing not 7 or 10 phase 2 clinical trials. You are evaluating the whole population with this mutation. And as soon as you have evidence, you get one application, and after a few months, the next one, and so on. But now look at the National Institute of Health, 2015. There was a meeting in Washington, and they reached the following conclusions. There is a growing concern that the results obtained from clinical research may not applicable to real-world situations. So randomized clinical trials, the gold standard, probably are not applicable. We need for more evidence to inform decisions that lead to improved, efficient, and affordable care. White quality evidence generated by conducting randomized clinical trials and disseminated through clinical practice guidelines is severely lacking. If you read the JAMA last week, there's a beautiful paper that indicates to you which is the percentage of increase in class 1A recommendations at both sides of the Atlantic. In the absence of evidence, because most of the recommendation, many recommendations came from observational studies of expert opinion, but this is your expert opinion, but probably it's not my expert opinion. Okay, so they said that the kind of trials need to provide medical evidence to support treatment decisions probably are not going to be made because simply the pharmaceutical companies are not going to support them, and the national governments are not going to support this type of studies. So the vast majority of clinical trials are too small. Look at these four bullets, okay? Because they are pretty tough. The vast majority of clinical trials are too small to provide sufficient statistical power to definitively answer clinical questions. They fail to address clinical treatment priorities and/or suffer from shortcomings in design and execution that limit their usefulness. Data from many clinical trials are not reported in timely and transparent ways. And finally, many findings published in peer-reviewed literature are fundamentally unreliable. And so what did they propose, particularly at Duke University? We need to change. We need to go to what they call pragmatic trials. The pragmatic trials, look, it's not to answer one question, a scientific question. Look at this. This is the main difference. The pragmatic clinical trials represent a new approach to engage more patients, clinicians, and sites to create a learning health system that reinforces high-quality health care. It's based in team-based care, electronic health records, the advantage in information technology and computer powered by informatics to create very large and complex set of data at very low cost. And please know, it's not to answer a scientific question, a very sophisticated question. They're designed for the primary purpose of informing decision makers regarding the comparative balance of benefits, burden, and risk of a biomedical or behavioral health intervention at the individual or population level. So it's not to inform to the scientific community, it's to inform to patients, clinicians, administrators, and policymakers. I'm going to skip a couple of them. Look, this is more or less the idea. Who developed the questions? This is randomized clinical trials. These are pragmatic trials. Researchers. Here are decision makers. Patients, clinicians, administrators. Probably, as if I am an administrator, I would like to know something that never will be performed in a clinical trial. What is the purpose? To determine the causes and effects of treatment. No, here is create generalizable knowledge, improve care locally, and inform clinical and policy decisions. What question does it answer? Can this intervention work under ideal conditions? This is the randomized clinical trial. No. Does this intervention work under usual conditions? And so on. You know, who collects the data? Randomized clinical trials are researchers. What is a study? A biological mechanistic hypothesis. In pragmatic clinical trials, as you can see, what is a study? Compares the balance between benefits and risk of health intervention at the individual or population level. What is compared the effectiveness at real-world alternatives. So you see it's a big change in the way to develop clinical trials. More or less, this is the idea. This is the pragmatic trial. This is all the patients. because the randomized clinical trial that's published in New England Journal of Medicine, its impact factor is pretty high, but probably only can apply the information of this clinical trial to how many? 20, 40, 60% of the patients? Do we read carefully the exclusion and inclusion criteria? So this is more or less the idea. To create a pragmatic trial is to improve the healthcare to general population. I know that this can be a lot of criticism, there are many criticisms. So what I present to you is that we need to improve, to develop more compounds faster, cheaper, and more effectively. So understand the drug properties, as I told you, for me is key, the phase 2 clinical stage. In phase 3, incorporate primary endpoints, as already mentioned, according to the mechanism of action. Focus on a specific subset of patients most likely to benefit, all versus niche population, using to identify these specific subsets of patients, imaging biomarkers, and of course using sample banking, replace sites by consortium, exchange information in a secure and auditable fashion, decrease the bureaucracy, and finally be familiar with new approaches for the design and analysis of clinical trials. And I just present to you something about pragmatic and adaptive clinical trials. So more or less are some of the changes that happen in very recent years in our field, okay?